Hello everyone. So this is the continuation report of Miss Christine Jan Sabdullah. By the way, I am Roslian C. Tantingco. Alright, amino acid metabolism. Amino acid forms metabolic intermediates by transamination using the amino group from other amino acids. So deamination, it is the removal of amino nitrogen as urea via or urea cycle. The carbon skeletons that remain after transamination, it oxidized via the citric acid cycle used to synthesize glucose gluconeogenesis. It is form of ketone bodies and or acetyl-CoA which may be oxidized or used for synthesis of fatty acids. So if naminaw mo sa previous reporter, nakasabot na mo sa kaning mga term na akong discuss. So amino acid may also be precursors of other compounds, purines and pyrimidines, hormones such as epinephrine and thyroxine, and neurotransmitters. So, next slide. So, balikan na nato ni siya. As you can see, metabolic reactions fall into one of two subtypes. We have catabolism and anabolism. Catabolism is all metabolic reactions in which large biochemical molecules are broken down to smaller ones. Catabolic reactions usually release energy. The reactions involved in the oxidation of glucose are catabolic. Next is anabolism is all metabolic reactions in which small biochemical molecules are joined together to form larger ones. Anabolic reactions usually require energy in order to proceed. The synthesis of proteins from amino acids is an anabolic process. Next, this is the process of how metabolism works. So in this stage, this is the stage of digestion. The large organic molecules of organic chemistry like proteins, lipids, and polysaccharides are digested into their smaller components outside cells. This stage acts on starch, cellulose, or proteins that cannot be directly absorbed by the cells. In stage 2, it is the release of energy. Once the molecules are broken down, these molecules are taken up by cells and converted to yet smaller molecules, usually acetyl coenzyme A, which releases some energy. So in the third stage, it is the energy stored. The released energy is stored by reducing the coenzyme nicotinamide adenine dinucleotide into NADH. This process provides the chemical energy necessary for the maintenance and growth of cells. Some examples of the catabolic processes include the glycolysis and citric acid cycle, the breakdown of muscle protein in order to use the amino acids as substrates, or gluconeogenesis, the breakdown of fat in adipose tissue to fatty acids, and oxidative deamination of neurotransmitters by monoamine oxidase. Here we have the summary. So these are the metabolic profiles of K tissues. So first we have the brain. Usually, neurons use only glucose as energy source since the brain stores only a very small amount of glycogen. It needs a steady supply of glucose During long fast, it becomes able to oxidize ketone bodies. When we say ketone bodies are produced by the liver and used peripherally as an energy source when glucose is not readily available. So next is the liver. The maintenance of a fairly steady concentration of glucose in the blood is one of the liver's main functions. This is accomplished through gluconeogenesis and glycogen synthesis and degradation. It synthesizes ketone bodies when acetyl-CoA or acetyl coenzyme A is plenty. When we say plenty, daghan. So it is also the site of urea synthesis. Another we have adipose tissue. It synthesizes fatty acids and stores them as triacylglycerols. Glucagon actives a hormone-sensitive lipase which hydrolyzes triacylglycerols, yielding glycerol and fatty acids. These are then released into the bloodstream in lipoproteins. When we say lipoproteins, are substances made of protein and fat that carry cholesterol through your bloodstream. Next is muscle. Muscles use glucose, fatty acids, ketone bodies, and amino acids as energy source. It also contains a reserve of creatine phosphate and phosphate, a compound with a high phosphate transfer potential that is able to phosphorylate ADP to ATP. Thereby, if mo produce kag energy without using glucose, the amount of creatine in the muscle is enough to sustain about 3 to 4 hours of exertion. After this period, the muscle uses glycolysis first and aerobically since it is much faster than the citric acid cycle, and later when the increased acidity slows phosphorotokinase enough for the citric acid to become not rate limiting in aerobic conditions. Next is the kidney. It can perform Next is kidney. It can perform gluconeogenesis and release glucose into the bloodstream. It is also responsible for the excretion of urea, electrolytes, and etc. Metabolic acidosis. When we say metabolic acidosis, is a clinical disturbance characterized by an increase in plasma acidity. Metabolic acidosis may be increased by the action of the urea cycle, since urea synthesis, which takes place in the liver, uses HCO3, thereby further lowering blood pH. Under these circumstances, nitrogen may be eliminated by the joint action of kidney and liver. Excess nitrogen is first incorporated in glutamine by glutamine synthetase. Kidney glutaminase then cleaves glutamine in glutamate E NH3, which the kidney immediately excretes. This process allows nitrogen excretion without affecting blood bicarbonate level. And last but not the least, the hormone control. Hormone control is mainly effected through the action of two hormones synthesized by the pancreas, insulin and glucagon. Insulin is a peptide hormone produced by beta cells of the pancreatic islets. It is considered to be the main anabolic hormone of the body. So just like what happened to Ivana, Alawi's sister, Simona, nasobrahan iyang insulin sa iyang lawas, then unsa nitabo niya is naluya siya, And then nag-agaw buhay man to siya before iyahang birthday. So, kaning insulin is delikado siya if masobraan siya sa imong lawas. Dapat average lang siya. So, insulin is released by the pancreas when blood glucose levels are high. After a meal, insulin stimulates glucose uptake by the muscle, glycogen synthesis, and triacylglyceride synthesis by the adipose tissue. It inhibits gluconeogenesis and glycogen degradation. So glucagon is a hormone that is involved in controlling blood sugar glucose levels. Glucagon is released by pancreas when blood glucose levels drop too much. Its effects are opposite those of insulin in liver. Glucagon stimulates glycogen degradation and the absorption of gluconeogenic amino acids. It inhibits glycogen synthesis and promotes the release of fatty acids by adipose tissue. So, to sum up, organ systems work together to accomplish their tasks. So, the different organ systems also cooperate to keep the body running. Next is the definition of terms. First, we have metabolic pathway. It's a series of chemical reactions in a cell that build and break down molecules for cellular processes. Next is UDP glucose. Uridine diphosphate glucose is a nucleotide sugar. It is involved in glycosyltransferase reactions in metabolism. Next is cellulose, a ketopentose, a monosaccharide containing 5 carbon atoms and including a ketone functional group. It has the chemical fructolysis refers to the metabolism of fructose from dietary sources. Next is epimerization. Is the conversion of a compound into an epimer, diglucose, and digalactose, partially or complete. So, mo niyang definition sa mga difficult terms. And that's all. Thank you.